My name is Tom Walter. I'm a corporate fellow at Waters, where I've been involved with developing HPLC columns for over 35 years. I chose the topic of UHPLC columns because 2024 marks the 20th anniversary of the introduction of the first commercial UHPLC system, the Acuity UPLC system. I was the director of the chemistry R&D group that developed the columns for this system and oversaw the development of additional UHPLC columns for many years thereafter. Today, I'll talk about the fundamentals, the column chemistries, and some recent applications for UHPLC columns. One note on nomenclature. The term UHPLC has been used to mean different things. For this talk, I'll use it to designate LC with columns packed with sub-2 micron particles designed to tolerate pressures up to about 18,000 psi. The name UPLC is a Waters trademark for systems and columns for UHPLC. First, let's briefly review the fundamentals of UHPLC. The idea of using the smallest possible particles for liquid chromatography has a long history. In 1941, Martin and Singe predicted that using very small particles and a high pressure difference would give the lowest height equivalent to a theoretical plate, which would mean that the bands of the separated analytes would be narrow. However, given the use of open columns and gravity flow, it wasn't possible at that time to put this idea into practice. It would be another 25 years before this vision would become experimentally feasible, when the earliest high-pressure LC experiments were reported. The first commercial HPLC systems had pressure limits of about 6,000 psi. These were initially used with columns packed with 50 micron particles, but the particle sizes gradually decreased, reaching 1.5 micron by 1995. However, the pressure limits of HPLC systems remain constant, so the use of sub-2 micron particles was limited to relatively short column lengths. This began to change in 1997 when Jim Jorgensen's group reported a capillary ultra-high pressure LC system with a pressure limit over 60,000 psi and demonstrated column efficiencies up to 330,000 plates for capillary columns packed with 1.5 micron non-porous C18 silica particles. Jorgensen used capillary columns to minimize frictional heating. Inspired by Jorgensen's research, Waters developed the first commercial UHPLC instrument, which was named the Acuity UPLC system. Unlike Jorgensen's capillary setup, this system was designed to work with 2.1 millimeter ID columns and had a pressure limit of 15,000 psi. The columns were packed with 1.7 micron porous hybrid particles that were C18 bonded, and initially only 50 and 100 millimeter lengths were offered. As predicted by theory, the minimum plate height was shown to decrease in proportion with the particle size. Also, the linear velocity giving the lowest plate height increases as the particle size decreases. Several instrument attributes were key to achieving the improved performance of UPLC. First is the increased pressure limit of the system, since column back pressure at the optimum linear velocity and constant ratio of length to particle size, or L over dP, increases as the square of the inverse particle size. At constant column length, this dependence increases as the cube of the inverse particle size. A second critical consideration is the extra column dispersion, which needed to be reduced in proportion to the peak widths. The detector sampling rates needed to be increased to ensure a sufficient number of points across the narrow peaks. And the thermal environment of the column needed to be semi-adiabatic to minimize radial temperature gradients, which can reduce column efficiency at high linear velocities. This was accomplished using a still air oven. The benefit of UHPLC over traditional HPLC is that it can provide faster separations without compromising resolution. Since column efficiency is proportional to column length divided by particle size, and the optimum flow rate is proportional to the inverse of particle size, at constant L over dP, analysis time is proportional to the square of the particle size. This is shown in these chromatograms, where the ratio of column length to particle size was held approximately constant, so that the resolution was maintained as the separation time was reduced from 10 minutes to about 1 minute. The flow rate was also increased proportionally with the inverse of the particle size. The combination of the threefold shorter column length and the threefold higher flow rate gives a ninefold faster separation for the column packed with 1.7 versus 5 micron particles. Comparing columns packed with 1.7 versus 3.5 micron particles, the separation is 4 times faster. And for the comparison of columns packed with 1.7 versus 2.5 micron particles, the separation is 2 times faster. Another benefit of UHPLC over HPLC is that it generally has reduced environmental impact, since the use of shorter analysis times and microbore columns results in reduced mobile phase and energy consumption. Comparing methods using a 2.1 millimeter column packed with 1.7 micron particles versus a 4.6 millimeter ID 5 micron column with the same L over TP, there's a 93% reduction in mobile phase usage. For the same column comparison, the energy consumption is reduced by 89%. Not only does UHPLC reduce the environmental impact of chromatographic separations, but the manufacture, distribution, and disposal of UHPLC columns have less environmental impact than that of conventional HPLC columns. We recently worked with My Green Lab to create ACT eco-labels for 42 Acuity and Cortex Premier columns. These labels may be obtained from the My Green Lab website. These are the first LC columns with ACT eco-labels. Now let's take a look at stationary phases for UHPLC. I'll start by describing the 4 different support particles that Waters offers. Each one has a specific purpose. Our ethylene bridged hybrid, or BEH particle, is designed for stability over a wide pH and temperature range. Charged surface hybrid, or CSH particles, are based on BEH particles that have been modified to add a positive charge. This provides excellent peak shape and loadability for basic analytes when using low ionic strength acidic mobile phases, such as those containing formic acid. It also gives increased retention for anions, such as ionized acids. High-strength silica, or HSS particles, are designed to provide high retention, particularly for reverse phase separations of polar analytes. These first particles are all fully porous. We also offer Cortex solid core particles, which provide higher efficiency, allowing for faster separations. As mentioned earlier, the first Acuity UPLC columns were packed with C18 bonded ethylene bridged hybrid particles. These particles, which were being offered for the first time, have several desirable features for use in UHPLC. First, they have excellent mechanical strength, so they can be packed and used with high pressures. Second, they have low silanol acidity, so they generally provide sharp peaks for basic analytes with minimal tailing. And third, they have exceptional base stability compared to C18 silica columns, as shown in this chart. This makes them rugged and highly versatile. Remarkably, today BEHC18 is still the most used UPLC chemistry. There are several reasons why it's beneficial to use a column that's stable over a wide pH range. First, varying mobile phase pH is the most powerful way to change selectivity for ionizable analytes. An example is shown here for representative acidic, basic, and neutral compounds. For the basic compound to be fully deprotonated, the pH needs to be 8 or higher, which is out of range for typical silica-based C18 columns. Second, peak shape is often dependent on mobile phase pH. In reverse phase chromatography, the best peak shape and loading capacity are achieved at pH values where the analyte is neutral. Besides acid-base equilibria, there are other equilibria that affect peak width. These may be pH dependent and so can be controlled by adjusting the pH. Another phenomenon that affects peak shape for some analytes is adsorption on metal surfaces in the column and HPLC system. Generally, adsorption decreases when the mobile phase pH is higher than 7. The final reason is that detection sensitivity often changes with pH, particularly when using electrospray ionization mass spectrometry. Here I compare the results obtained at pH 10.6 and 2.9 for a mixture of acids, bases, and neutrals. The compounds shown in blue are bases, those in red are acids, and those in black are neutral. As expected, the acids are better retained at low pH, where they're not ionized. Conversely, the bases are more retained at high pH, where they're not ionized. The retention of the neutrals is largely the same at both pH values. In order to optimize separations, we recommend investigating both high and low pH mobile phases during method development. When we developed the 1.7 micron BEHC18 stationary phase, with the focus being on achieving high efficiencies and mechanically stable columns, we didn't compromise on reproducibility. Even though we used more complex synthesis processes, with a new particle and trifunctional bonding chemistry, we achieved excellent batch-to-batch reproducibility. This chart shows the chromatographic QC results from over 19 years of production, demonstrating relative retention RSD values of 3.2% and lower. This is from a UPLC version of a test originally developed by Uwe Neue. The RSDs are comparable to those for the most reproducible silica-based C18 columns. This slide shows a representative separation for an acuity BEHC18 column using a modified version of the Neue test. Note the excellent peak shape for the basic compound amitriptyline, peak 7, with a tailing factor of only 1.15. There are currently 8 different chemistries available in 130 angstrom BEH particles, 4 designed for reverse phase chromatography, 3 for hydrophilic interaction chromatography, and 1 for mixed mode reverse phase anion exchange chromatography. All of these materials have greater stability in basic mobile phases compared to equivalent silica-based bonded phases. An interesting phenomenon affecting the peak shape in reverse phase chromatography became apparent when using low ionic strength acidic mobile phases, which are generally preferred with mass spectrometry detection. The most common example is 0.1% formic acid. With these mobile phases, high tailing factors are seen for both ionized acids and ionized bases at relatively low mass loads. This was seen when using high purity C18 silica columns and also for a polydivinyl benzene column. So it doesn't appear to be caused by ionized silanol groups, but rather by electrostatic repulsion between the charged analytes when they are adsorbed on a stationary phase. We found that we could mitigate this problem by applying a low concentration of weakly basic groups on the particles, then bonding with C18 or other groups. This was named Charged Surface Hybrid, or CSH technology. The chromatograms compared the loading behavior for the basic analyte quetiapine for a CSH-C18 and a BEH-C18 column, using mobile phases containing 0.1% formic acid. On the BEH-C18 column, the quetiapine peak shows typical overloaded peak shapes when injecting several hundred nanograms on a 2.1 by 50 millimeter column. With the same mass loads on the CSH-C18 column, no overload was observed up to an injection of 1,000 nanograms of quetiapine. In addition to CSH-C18, 2 other bonded phases are available based on the same charge-modified hybrid particles, a pentafluoropropyl material and a phenyl-hexyl stationary phase. These bonded phases were chosen to provide selectivities that differ from each other and also from that of BEH-C18. This is demonstrated in the chart shown here, which compares the retention factors determined for 20 acids, bases, and neutrals on each of the CSH chemistries to the retention factors on a BEH-C18 column. The relatively high scatter shows the selectivities are quite different. This may be quantified using the selectivity factor S, which is calculated from the correlation coefficient. The S value of 49 is high relative to similar comparisons between conventional reverse phase columns. Now let's take a look at the high-strength silica, or HSS, chemistries. The HSS particles were developed to tolerate the high pressures needed for UHPLC. HSS has a higher surface area than the 130 angstrom BEH and CSH particles, which tends to give greater retention. It also has a higher concentration of surface silanols relative to BEH and CSH particles, which can be useful to retain polar analytes, particularly those that are basic. The 5 HSS chemistries shown here provide alternative selectivities to the BEH and CSH chemistries. Here I compare the separation of a mixture of 7 polar analytes using BEH-C18 and HSS-T3 columns. The T3 stationary phase is an intermediate coverage C18 bonded phase designed for use with 100% aqueous mobile phases. The mobile phase used for these separations was 98% aqueous, with 2% acetonitrile. The greater retention provided by the HSS T3 column is evident, and it's particularly beneficial for the early eluting compounds. The greater retention is due to the higher surface area and the greater silanol concentration of the HSS particles. This example shows the selectivity differences for basic analytes between the three C18 phases available on HSS particles. The HSS T3 material has a reduced C18 coverage versus HSS C18, and you can see the increased retention this gives. The HSS C18 SB material not only has a reduced C18 coverage, but is not end-capped, resulting in a relatively high concentration of silanol groups. The bottom chromatogram shows the effect this has on the retention of these basic compounds. Here I show a comparison of separations obtained for the 12 different acuity BEH, CSH, and HSS reverse phase chemistries. The analytes are naturally occurring polyphenols found in plants, and 4 of them have carboxylic acid groups. The chromatogram showed differences in retention and peak spacing. This is particularly evident for peaks 3 and 4, which elute in a different order on several of the columns. It's notable that the CSH columns show the greatest retention for these analytes, which is expected since most of them are acidic. A relatively recent addition to our UHPLC column portfolio is a mixed mode reverse phase anion exchange stationary phase named Atlantis BEH C18AX. This is based on a 95 angstrom version of our ethylene bridged hybrid particles, which has a surface area 50% greater than that of our 130 angstrom BEH particles. The C18AX stationary phase was optimized for retaining polar acids across a wide mobile phase pH range. It was also designed for reproducibility and pH stability, addressing 2 of the shortcomings of existing mixed mode materials. These columns are also compatible with mass spectrometry detection, showing minimal ion suppression or enhancement. Here I compare the selectivity of 4 different columns for the separation of a mixture of polar acids, bases, and neutrals. The mobile phase was 100% aqueous and contained a pH 3 buffer. The largest differences are seen for the 2 bases, nicotinamide and procainamide, and for the acid adenosine monophosphate. As expected, the Atlantis BEH C18AX column gave the greatest retention of the acidic analyte, peak 5. It also gave the least retention of the basic analyte, procainamide, peak 4. In contrast, the HSS T3 column gave the greatest retention for the 2 basic analytes. These trends are consistent with the characteristics of the stationary phases. As mentioned earlier, Cortex columns are packed with solid core particles. This chart compares Van Deemter curves for a 1.6 micron Cortex C18 column and a 1.7 micron BEH C18 column. The minimum plate height for the Cortex column is 30% lower than that for the BEH C18 column, due to smaller B and C terms. But despite the smaller particle size, the Cortex column has similar back pressure as the BEH C18 column. There are 2 factors that account for this. First, the Cortex particles have a narrower particle size distribution than the BEH particles. And second, the interstitial porosities of the Cortex columns are higher than that of the acuity BEH columns. An example of an application that benefits from the increased efficiency of Cortex C18 columns is the separation of synthetic cannabinoids. Here I compare the separation of 10 analytes using BEH C18 and Cortex C18 columns. Peaks 5 and 6, 7 and 8, and 9 and 10 are not fully resolved using the BEH C18 column, but are better resolved using the Cortex C18 column. This is especially important for peaks 9 and 10, which are isobaric, meaning they have the same mass. To accurately quantitate them, they need to be resolved chromatographically, because the mass spectrometer can't distinguish between them. The higher efficiency of 1.6 micron Cortex columns can be used to obtain faster separations. Here I show gradient separations of 7 sulfur drugs on a 1.7 micron fully porous C18 column and on a 1.6 micron Cortex C18 column. The top two chromatograms show the results obtained using the same gradient time and flow rate. As expected, the Cortex column gives a greater peak capacity, 218 versus 178. In the bottom chromatogram, the flow rate was doubled and the gradient time halved, producing a peak capacity that is similar to that of the top chromatogram, but with the separation achieved in only half the time. There are 7 different chemistries in the Cortex column family. In addition to Cortex C18, we have Cortex C18+, which uses bonding chemistry similar to that of CSH-C18 to achieve the best peak shapes and loadability for bases when using formic acid mobile phases. Cortex T3 uses an approach similar to that of HSS-T3 for enhanced retention of polar compounds. Cortex Shield RP18 uses bonding chemistry similar to that of BEH Shield RP18 and offers different selectivity, particularly for bases. Cortex C8 gives reduced hydrophobic retention, and Cortex Phenyl gives enhanced selectivity for aromatic analytes. Cortex Hillic is an unbonded particle and is intended for separating highly polar compounds using hydrophilic interaction chromatography. Here I compare the selectivity of the 6 reverse phase Cortex chemistries for a mixture of acids, bases, and neutrals. The bases are designated by the blue text, the acids by red text, while black text is used for the neutrals. The S values under the names of the columns indicate the selectivity difference relative to Cortex C18. The largest S values were obtained for the Cortex C18+ and Shield RP18 columns. Both of these show reduced retention for the bases relative to the Cortex C18 column. Now I'd like to briefly discuss columns for hydrophilic interaction chromatography, or Hillic. Hillic is a complementary chromatographic mode to reverse phase chromatography that may be used to improve the separation of very polar analytes. This is achieved by using a polar stationary phase in combination with a mobile phase that has a high concentration of an organic solvent, typically acetonitrile, and a low concentration of water. In Hillic, analytes elute in order of increasing hydrophilicity. Here I show a comparison of the differences in retention and selectivity for the 4 Waters UHPLC Hillic stationary phases. The Atlantis BEH Z Hillic column provides the greatest retention of all 5 analytes, followed by the BEH Amide column. The BEH Hillic and Cortex Hillic columns, which are based on unbonded particles, give the least retention for these compounds. The high efficiency of the Cortex column is evident from the narrower peaks. A recent focus of column development has been to address the issue of analyte adsorption on metal surfaces. In 2020, we introduced modified column hardware in which a hybrid composition containing bridging ethylene groups is applied to the metal surface. This was named Max Peak Premier technology and has been incorporated into both column hardware and the flow path in LC systems. The chromatograms compare separations of adenosine mono, di, and triphosphate using a Max Peak Premier column versus a conventional stainless steel column, both run on an Acuity Premier system. Improvements in peak width and peak shape are evident for ATP and ADP. Now let's take a look at some recent examples of applications of these columns. The literature on UHPLC now encompasses more than 35,000 articles. The applications range widely from chemistry and biochemistry to pharmacology, agricultural, and medical studies. I'll present 4 examples to illustrate how UHPLC is being used for separations of small molecule pharmaceuticals, cellular metabolites, and environmental contaminants. The first example takes advantage of the ability of UHPLC to provide fast separations. This is beneficial for applications that involve only one or a small number of analytes, but for a large group of samples. Examples include adsorption, distribution, metabolism, and elimination studies for small molecule pharmaceuticals. By using short columns and high flow rates, run times less than a minute may be achieved. A recent application note describes a high-throughput LCMS approach for ADME studies. A 30 millimeter long 1.6 micron Cortex T3 column was used with a high flow rate, 1 mil a minute, and a sub 1 minute gradient. The peaks were detected using tandem mass spectrometry. As shown here for 5 different small molecule pharmaceuticals, relatively narrow and symmetrical peaks were observed, making integration straightforward. This method was used to quantify the concentration of the compounds when they were incubated with microsomes to determine their metabolic stability. This methodology is suitable for labs that need to rapidly analyze large numbers of samples from ADME studies. The second example illustrates the use of UHPLC to rapidly develop methods for separating small molecule pharmaceuticals for force degradation studies. Method development was carried out using a systematic screening protocol that uses 6 different stationary phases, 2 different organic solvents, and low and high pH mobile phase additives. Altogether, 24 combinations need to be tested. The benefit of using UHPLC over HPLC for method development is that the time to complete the screening runs is shorter. For the example I'll show, the runtime per injection was about 10 minutes. So with 3 injections per combination, the total time was 12 hours. If the same work was done using a 3.5 micron HPLC column with equivalent length to particle size ratio, the total time would be 48 hours, which is 4 times longer. As described in the application notes cited here, the systematic screening protocol was used to develop a method for analyzing a force degradation sample of a PROTAC therapeutic. The results obtained using 6 different UPLC columns with the same mobile phase gradient are shown. The results for the other mobile phase combinations are described in the application note. The best separation overall was achieved using the Acuity Premier HSS T3 column. Here I show the final optimized method using the Acuity Premier HSS T3 column with an acetonitrile gradient and 0.1% formic acid as the additive. The peaks were detected by mass spectrometry. Good peak shapes were obtained for the parent compound and all the degradants with a total runtime of less than 10 minutes. The structure of the parent compound is shown. It has 2 active domains connected by a linker. One domain binds to the target protein and the other to an enzyme that labels it for proteolysis. This is an example of some of the complex small molecule pharmaceuticals that are currently being developed. Because of their complexity, high peak capacity separations are needed to resolve all the degradants. Another important application area that benefits from UHPLC is metabolomics. A number of studies have shown that UHPLC can not only provide faster methods, but also higher resolution. In the article cited here, an HPLC method was compared to a UHPLC method that is 2.7 times faster. Not only was the UHPLC method faster, but it provided more than 2 times the number of peak identifications. With these compelling benefits, it's not surprising that a recent analysis of the separation methods in the public data repositories Metabolites and Metabolomics Workbench found that 64% of the methods use columns packed with sub-2 micron particles. The hybrid surface technology that I mentioned earlier has been shown to greatly improve the peak width for a number of metabolites. The examples shown here demonstrate the improvement seen for 2 acidic lipids, phosphatidic acid and lysophosphatidic acid, with the best results seen when using HST in both the column and the system. For these examples, CSHC18 columns were employed, and negative ion electrospray mass spectrometry was used for detection. 2 important groups of metabolites are those associated with glycolysis and the pentose phosphate pathway. These compounds are very polar, so HILIC is used. In addition, many of these analytes are phosphorylated, making them challenging for conventional UHPLC columns and systems. An optimized separation of 27 metabolites using an Atlantis Premier BEHC HILIC column and an Acuity Premier system is shown here. The mobile phases contained a pH 9 ammonium bicarbonate buffer, so it's important that the column is stable at this pH. The peaks were detected using a tandem quadrupole mass spectrometer, some with negative ion electrospray and some with positive ion. The 2 isobaric pairs are well resolved, that is, peaks 5 and 6 and 10 and 11. Good peak shapes were seen for all the analytes. The last examples I'll show involve the determination of per- and polyfluoroalkyl substances, or PFAS. These are a group of long-lasting industrial chemicals that are widely used because of their unique chemical properties. Unfortunately, they've been found as contaminants in water, soil, and animals, including humans. Exposure to some of these compounds has been linked to harmful health effects in people, and regulations have been put in place to monitor and limit their concentrations. Quantitation of PFAS can be challenging because thousands of different compounds exist. They are generally found at part per billion levels, and background contamination is a problem because many of these compounds are found in labs. UHPLC coupled with tandem mass spectrometry offers sensitive, high-resolution methods for these compounds. The application note cited here describes a UHPLC MS method for quantifying 46 PFAS compounds in drinking water. An Acuity Premier BEHC18 column was used with a 17-minute methanol gradient. Good peak shapes were observed for all of the analytes, and in some cases, the linear and branched isomers could even be resolved. My last example is for the separation of ultra-short chain PFAS, including trifluoroacetic acid, TFA, perfluoropropionic acid, PFPRA, and perfluoropropane sulfonic acid, PFPRS. As shown in the top chromatogram, TFA elutes in the void volume when using a BEHC18 column, and the PFPRA peak is close to the void. When an Atlantis BEHC18X column is used, however, the TFA peak is separated from the void, and the PFPRA peak is much better retained. This mixed mode column retains these ionized acids by both anion exchange and reverse phase interactions. The gradient method shown here is also capable of separating long chain PFAS. This allows the ultra short chain and long chain PFAS to be analyzed in a single injection. To wrap up, I've reviewed the benefits of UHPLC and the current state of UHPLC column technology. The main benefit of UHPLC is higher efficiency per unit time, allowing faster separations without sacrificing resolution. Reduced energy and mobile phase consumption is a benefit that's becoming more relevant with current initiatives to reduce the environmental impact of lab operations. The wide range of column selectivities currently available enables separations of many different analytes. And the recent introduction of inert hardware offers improved performance for compounds that adsorb on metal surfaces in the column and LC